I'm really pleased now to introduce the next panel in our program. The title is Reinventing Research, AI in the Future of Health Innovation. Now, according to your program, Dr. Suchi Saria was going to be our moderator, but unfortunately, she is having some traveling difficulties, something I'm sure everyone in this audience has experienced in the past. So Dr. Peter Lee, President of Microsoft Research, and also a two-time graduate of University of Michigan, just like me. So a little leverage there, has agreed to take Dr. Saria's position in this panel. So Peter, I leave it to you.
Thank you. We're on a panel on reinventing research, AI, and the future of health innovation. And so we're really going to try to look at the trends in the future, what it means for innovation, what it means for research. I have several reasons to be very nervous about this. As Eva described, Suchi really put a lot of work into organizing this panel, and I think it was exceptionally well prepared. And there's no way now that we can be as organized, but we'll do our level best. The second reason to be nervous is, of course, the session this morning was just very hard to follow. And then Aviv's talk was incredible. So hopefully we'll not waste everyone's time here. And then the third reason to be nervous is my wife is in the audience, and I'm always a little bit more nervous when that happens. But we will do our best. The full biographies of our panelists are in the packet. Just in absentia, Suchi Saria is the John C. Malone Professor of Computer Science and also of Applied Mathematics and Statistics at Johns Hopkins University. And very notably, she is the founder and CEO of Bayesian Health. And I think in 2022 was named as one of the most significant figures in artificial intelligence. And in 2022 was very famously the developer of now a widely deployed AI model that has made double-digit reductions in sepsis. And so just incredible there. But we do fortunately have panelists here. We have Dr. Fiona Marshall, the president of Biomedical Research at Novartis, Dr. Lovisa Afzelius, general partner of Flagship Pioneering, and also the founding CEO and co-founder of Metaphore Biotechnologies. I'm sure we'll hear more about that today. And then at the far right, your far left, Dr. Paul Harris, the Vice President for Research Informatics, and also the Director of the Vanderbilt University Medical Center Office of Research Informatics. And so we'll just dive right in. And now I'm going to jump out of my moderator role and give my opening remarks. When we think about the future, we actually can go back into the past because for many years now, actually for a couple of decades, we've had a dream. And that dream has been to use machines to allow us to somehow just write down what we want. If you're trying to design a new molecule, trying to develop a new drug, can we just write down what we want and then give it to this magical AI model, and then out pops the molecule design that we want? How can we do that? And as we look to the future, it's looking increasingly like this future may actually be feasible. How? There are many, many brilliant approaches that are being taken here. And actually, Aviv's talk this morning gave, through case study examples, some explanation of several approaches. But I thought it might be valuable here just to say at a very high level, maybe the simplest, most naive approach that actually seems to be working in practice. And it's just one of several approaches now that are underway. While we have always had, for a couple of decades, computational methods to simulate proteins and molecules at the atomic level, at the quantum, with quantum precision, these simulators, as well as the lab observations, have always suffered from being impractically slow. And so it is still important, and in fact, it's a hallmark of scientific progress that the rise of computational science, all of those classic Fortran codes to do precise simulation of molecules, of materials, of chemicals, they exist, and we know that they're grounded in the reality of Schrödinger's equation and of everything we know about physics. The only problem has been that as a way to actually discover and design new molecules, they've just been impractically slow. And famously, even very small biomolecules, to fully simulate them using those Fortran codes on the world's largest supercomputers could take years, sometimes even in the millions of years. Now, what has changed? Well, despite the fact that these simulation codes are so impractically slow, they do run and they do produce precise data. And that data now is valuable because it rarely represents, oftentimes, perfectly labeled data that can be used to train AI models. And so in the era of generative AI, we now have the kind of magical ability to use various training methods to take the output of lab data and simulator data through training to produce an AI model. And the fact that this actually works has just sparked an avalanche of excitement in the research field as well as in the commercial venture capital field. And literally a month doesn't go by where a CNS journal, a Cell, Nature, or Science journal, doesn't have one or more articles showing some example of this effect. And just if I could brag for Microsoft Research, we participated in this as well. This is a paper by Frank Noy and Chris Bishop and team at Microsoft Research that was on the cover of Science 2 months ago. But we are far from alone. There are hundreds of great laboratories around the world that are pursuing some variants of this type of cycle and increasing the optimism that we may be able to achieve that original dream. And so as we listen to our panelists here, I think we're going to hear different shades of some element of this type of pattern and what it actually means in the real world and what it might mean in the future. So I won't get into that detail, but instead what I thought I would do is the potentially foolish thing of trying to predict some elements of what we might see in the future. And foolishly, I'm going to make predictions going out 2 years from now, 5 years from now, 10 years from now, and 20 years from now. And so we'll see how this goes. All right. So in 2 years, and some people will complain that this is already here, this is happening. But I think we will see that the use of AI to design candidate biomolecules in the process of drug development and drug discovery will become standard practice, not only in biomedical research, but in chemistry and chemical engineering and in material science. In 5 years, harnessing real-world data. Of course, today we are seeing the rapid rise of highly personalized and targeted therapies, particularly in the cancer domain. But within 5 years, I think this will spread well beyond cancer to really just hundreds or maybe even thousands of different medical and disease conditions. In 10 years, I suspect we'll hear more about this from both Fiona and Lovisa. I think we're going to see practical closed-loop discovery systems, AI-mediated laboratories that run the whole cycle of hypothesis generation, experimental design, wet lab automation, analysis of wet lab results, reanalysis of hypotheses, and so on. And then in 20 years, I was actually talking to Eric Topol. He predicts this as in 10 years, but I think that's a little optimistic. In 20 years, we'll have something similar to what Aviv was describing this morning in her lecture, something similar to a very comprehensive emulation of whole cell dynamics. Now, that all seems good, and you would expect a techie like me to be very optimistic about things, and so I would say these are the optimistic projections. I thought it would be fair, though, to also make predictions on these timescales of things that maybe will also happen that might not be as good. And so we are actually already in the biotech winter, and there has been way more venture capital invested in biomedical molecular design startups than the total valuation of all those startups combined. And so I think there's going to be a bubble bursting, and that is going to dampen enthusiasm. It will not dampen the actual science, but I think enthusiasm may dampen. In 5 years, I think there is a reasonable chance that up to 40 nations in the world will ban or move to ban the use of AI in biomedical research out of concerns for safety and potential rise or acceleration of bioterrorism threats. I think it's possible that we'll have a first Nobel-worthy discovery with no human in the loop. And I think that will be a crisis. I don't know if Eva will know how to get the AI on stage to talk about it. And then in 20 years, I think it's quite possible that there will be discoveries made, possibly in partnership, possibly not in partnership with humans, that will be so complex that all we'll be able to do is to validate and verify that they are correct discoveries, but we won't understand them. And I think that will be a crisis, a psychological crisis for all of us in the research world. So with that, I am now going to pass off to Fiona.
Okay, thank you, Peter. So, yes, I'm Fiona Marshall, and I lead biomedical research at Novartis. Our goal is to really treat diseases in new ways now by trying to target the underlying cause of disease. And in doing that, to try and create medicines that don't just treat the symptoms, but really try and put diseases into long-term remission or ultimately actually cure the disease. And try to do this for patients around the world. Of course, this is an enormous challenge. And for any given program, as you probably know, I mean, we usually make thousands and thousands of molecules and test them preclinically. And we're fortunate in research if even a few of those actually even make it into clinical testing. So we really need to improve the probability of success. And of course, AI, I think, has the potential and is already demonstrating the ability to improve these odds. But I have to say, it's not a magic wand. Maybe I'm a bit more skeptical than Peter is on how it's really going to deliver. There's many steps in going from creating a molecule to actually making and approving a new medicine. And I think AI is going to be really effective at certain of those steps, but I'm not convinced that all of those steps could be done by AI. So what we're trying to do at Novartis is really evaluate AI through those steps across both research and development and identify where it can have the greatest impact. And I want to share 3 real examples of what we're doing now to sort of maybe ground us in the here and now and then help us to think about what the future would bring. So obviously the first step, and I'm just putting up this one slide, the first step in drug discovery, of course, is to design a new medicine. And it's first of all starting with patients to really understand their disease, how they think about their disease, and where the unmet need is. And then we need to identify and validate biological targets that can modulate the processes underlying the disease that will then provide that benefit needed. And what we try to do is to focus on where the clear gaps are in healthcare, where patients are lacking effective treatment options. Now, it's no secret that biopharma, and this includes venture capital finances, tend to crowd into the same targets in the same areas all at the same time. And we've certainly seen that very clearly in the obesity area recently. So for me, one of the real opportunities that AI can help us is to maybe move away from the crowd and discover new targets faster that can really unlock what are truly innovative therapies rather than bringing incremental benefits of more of the same. Now, one of the areas we're really focusing on at the moment at Novartis is kidney diseases. They actually, I think, have been understudied, and really there's a high unmet need, both from the very common chronic kidney disease through to some of the rarer kidney diseases. Now, the most common inherited kidney disease is autosomal dominant polycystic kidney disease, or ADPKD for short, and that is marked by the growth of cysts that over time lead to kidney failure. Now, our teams have worked on this for many years, really trying to identify druggable targets. We know the genetic basis, but how do we approach that with targets? And we've had limited success. So this was one of the ones we decided to apply AI to, and really to conduct large-scale tests where we're simulating, turning genes on and off, looking for potential involvement in the disease. And what we do is really analyze all of the data we can find available, whether that's scientific literature, obviously the human genetics, data from millions of single-cell experiments, as well as our own internal clinical data. And we rapidly generated a list of 72 candidate genes just based on pure computer modeling and AI, but then tested those out in wet lab experiments. Now, a key here is to have the right wet lab experiments to complement your AI. And in this case, we had human organoid models from patients who have ADPKD. So these really essentially are, and there's a nice, this is the picture here, is actually, you can see that the cells actually form a lot of the cells that you would have within the kidney structure, but they also develop the cysts. So a really good model system to test out the AI hypothesis. And from this, in under a year, we were able to identify 5, what you might call promising or even validated targets in this in vitro model that are now moving forward. So moving on to the next area, once we identify a target, the next stage is the generative AI. It's actually finding the therapeutic and to create the molecule that can become a medicine. Now here, generative AI, and I'm just talking now about low molecular weight compound type drugs, really helps us to explore a vast chemical space and design quality molecules. For a compound to be suitable as a drug, it has to balance huge numbers of different properties related to target biology, the pharmacology, chemical properties. And what we're trying to do, in this case, I'm just giving you an example of where we try to get an oral treatment for neurodegenerative disease. And these, of course, we have to design compounds that can get into the brain, but still be a good safety overall profile. So one of the examples I'd like to tell you about is in Huntington's disease. It's a devastating neurodegenerative disease, and progress has been slow. But here we used AI to generate, in this case, 15 million chemical structures, which we predicted could have activity at our target. And that was then narrowed down through various analysis down to only 60 molecules that the chemists then actually made that we thought would have the right profile. We continued then to refine those properties, again, still using digital models, and that included one for brain penetration. And this graph here you can see is the percentage pass rate. What you can see greatly increased over time once we started using AI, and we were able to make a lot smaller number of molecules as we went forward. And this has actually resulted in us being able to design a potent, highly brain-penetrant chemical scaffold that is actually now advancing through to late-stage optimization. Now, final example really is in the safety area. We can only progress a molecule if it has full safety. So one of our foundational resources at Novartis is Data 42. This is a vast data lake, and it includes 30 years of clinical data, including real-world evidence as well as preclinical data. And we use Data 42 to train AI models on thousands of toxicology and safety signals. And that comes from both successful and unsuccessful programs across all different disease areas. Now, as you might expect, some classes of compounds are associated with cardiac toxicity, and we really want to avoid this at all costs. So it could be abnormal rhythm, heartbeats, all sorts of cardiac toxicology. So using AI and the data from Data 42, we identified certain profiles that allow us to predict the potential of compounds for cardiac toxicity. And we're using this now really to profile compounds, and we had a very promising compound that ticked all the other boxes, but unfortunately came up, according to AI, as having cardio safety risk. Well, the team wanted to take it forward anyway, just in case, and what a surprise, actually it did have some cardio safety, so was then later terminated. So now the teams are more convinced that it makes sense to believe the AI prediction and not waste time, go and get another molecule. So you can see this example now. One of the things is, of course, persuading people to buy into this strategy. But really deciding when to stop a molecule or a program, to me, is notoriously difficult. It's one of the things where we can really deliver success very quickly, and AI can help tell us when to do that. This type of approach can also reduce the need for animal models, which I think is a priority for all of us as well, but most important, getting the highest safety molecules going forward to the clinic. So, I said at the start, AI, it's not a magic wand, but I think we are seeing some key areas where it really is having an impact, and we're going to sort of do even more of those. We do have molecules, so in 2 years, we do have molecules designed primarily through AI, but with humans in the loop. They are advancing through our pipelines, but there still are many challenges and headwinds that do slow us down. I still think understanding human biology remains incredibly complex, and translating the preclinical data, whether it's from in vitro assays or models into clinical studies, that still has to be done carefully. And there are many disease areas where the disease progresses quite slowly, so we still have to conduct really long-term clinical trials. But thank you for allowing me to share this opportunity to take a peek behind the curtain of what we're doing at Novartis and what we're trying to do to accelerate our work to advance new medicines. And over to you, Lovisa.
Super. Do you mind passing the clicker?
Fiona, there'll be a lot to follow up on, but we'll follow the pattern of the earlier panel. We'll each make some opening remarks, and then there's some prepared questions, and then we'll open up to the audience. So, Lovisa, you're next.
Thank you so much. So, good afternoon, everyone. My name is Lovisa Afzelius. I'm a general partner with Flagship Pioneering. Flagship Pioneering is a venture creation firm. It's probably mostly known for having founded Moderna, but we actually have founded about 100 companies since its inception in 2000. And I joined the firm back in 2020 with a remit to originate and build new companies. So I have a team of theoretical and experimental scientists, and together we come up with new ideas of what could create real value to patients and also stay in that non-adjacency space from what has been done before. My PhD is in computational chemistry, so that's why most of the companies that I founded, is in that interface between computational chemistry and biology. And it's interesting to see that although it was like 25 years since one wrote that PhD thesis, it's still so that the concept of the problems that we actually want to solve are many times the same. But it's just amazing to be in this time now when the tools, thanks to AI, allows us to just address them in ways we have just never been able to do before. So a couple of examples here. Alterna is our tRNA platform where we use machine learning to engineer tRNAs with the ability to read through premature termination codons. And in doing so, we have the potential, with a single medicine, be able to address thousands of ultra-rare and rare genetic diseases that share that premature termination codon. In a priori, we are building variant resilient antigen designs so that we can elicit an immune response that is protective both today but also in the future. And in Prolog, we're using knowledge graphs and we're using large language models to actually be able to leverage evolution by identifying therapeutics from the viral proteome. And in, for example, Contactome, we are using agentic AI to be able to understand what cell-to-cell contact may actually have the greatest relevance in disease. So as the one company I wanted to spend a little bit more time on is Metaphore. And the way our model works basically is that I stay on as CEO for these companies up until about its Series B, where I hand it over to an external CEO to really build the greatest version of the company. So as of the beginning of this year, I'm really proud that Angela Wang has taken over as CEO for Metaphore. So I'm presenting on her behalf on what we worked on since early 2020. And it's fantastic to see someone come directly from the executive leadership team of Pfizer to take on a platform like Metaphore, because I think it speaks volume to the science and what we can do with a platform like that. So with regards to Metaphore, what we believe that we have done is we have created a sequence to function generative AI model. And why this is important, and why I say function, is because all antibodies, or 95% of antibodies out there today, they actually block. They are blockers, they are antagonists. But really, if you think about biology, biology is about function. It's about driving downstream pathways and being able to control how those interact with each other, how to actually get to bias signaling, how to be able to have multiple different targets, cofactors, actually come together, and in that drive, that synergistic effects that you're looking for. So the question then becomes, why has it been so hard to systematically design antibodies that drive function? And I would say that one of the reasons to that is our starting points, because models are never better than the data that we build them from. And if we look at the models out there for generative AI molecular design, they are all based on crystal structures. It is really a structure where you may have done an internal truncation in order to get it out of the membrane, or that you separate it from its cofactor. It's in a setting which is very static. But I think most importantly, there is no connection between the bioactive conformation and the function that it's anticipated to solicit. So what if you instead could use living systems to solve for function and use that information to build your models? Because if you use such a cellular system, then you are in that native environment where you have all the cofactors coming in and actually be able to engage, and you understand what are the dynamic states that drive your function. So that's what we set out at Metaphore to do. We have built a high throughput, high or tight-seq, sort of high fidelity multiplex system that has allowed us to do deep mutational scanning. So basically testing hundreds of thousands of protein variants and understand how protein-protein interaction, how this single mutation changes function. And in doing so, we can build maps of that have very high, almost like single amino acid resolution functions. And now with these maps, we have built new AI in the sense that we first built a model we call Resolve that allows us to take this deep scanning data and use that data to parameterize our protein folding such that we can actually get to very explicit epitope-paratope interaction and mimic and extract out what are the key interaction features that are required to drive the downstream function. Then we have our Imagine models, which are basically novel diffusion models that allows us to populate those features with novel sequences and go very, very broad in the sequence space. And then simultaneously, we've developed novel models, evolved models that are protein large language models that are built off proprietary and public data sets that allows us simultaneously model in for developability so that we, in the end, can instantly get to molecules with the desired function as opposed to binding and with the development properties that we're looking for. And I will now speed through just one example where we applied it because I think the outcome data always speaks more than the actual theory behind it. And this is an example of a collaboration we have with Novo Nordisk, a $600 million collaboration in obesity in the GLP-1 receptor space, where we first showed that we could rapidly move from a peptide agonist into an antibody agonist, and in less than 3 months have a 10-fold more potent molecule than best-in-class drugs by using the largest surface area of the antibody to drive bioactive conformation. We then showed that we could generate these antibody or amino acid maps so that we could design in the multiple selectivity for multiple receptors in a single epitope and address that with a single paratope of the antibody. So basically multi-targeting several different GPCRs with a single paratope, and do that with absolute sequence diversity. All the ones in blue are hits that have no sequence similarity, and the ones in red are sequence similar. And just finally, just showcasing, we've also been able to really design in bias signaling so that in the context of GPCRs and GLP biology, wanting to increase the delta between cyclic AMP and beta-arrestin so that you can drive lesser side effect profile from a gastrointestinal side point. And we're able to do so very successfully. So that's sort of just an example of how we can use the new AI methodologies to really start addressing functions in a very sort of explicit way, which is currently not available from generative models.
Yeah, fantastic. You know, I think between your two comments and Aviv's lecture, there's probably never been more hardcore biology on the stage for this annual meeting than today. And so we'll probably continue. So Paul, you're next.
Thanks. So I'm Paul Harris. I'm a professor of biomedical informatics at Vanderbilt, and I run a team that supports research informatics. So we're always looking for new platforms to either build or adopt, or service lines where informatics can play a role in supporting the whole research enterprise. So either hypothesis generation or better, cheaper, faster. When we started thinking about this with respect to generative AI, which I'll talk about today, we started thinking about this from the perspective of, we need to think about some pilots that could use this technology to support that mission, but not just thinking about the pilots. Go ahead and concurrently think about the deployment. Do we have platforms that if we develop this workflow or this particular asset, that we can drop in for real-world scalability and robust use. So I'm going to talk about 3 of those today: the All of Us program, Research Match, and REDCap. All of Us is a large NIH-funded program that's supporting this notion of a consented cohort, where individuals from across the country can donate their health information, their EHR data, their genetic information, their wearable data. They take surveys, there are physical measurements, and there are multimodal data that's collected on these individuals for the purpose of handing that over to real-world researchers across the globe who can then use it for hypothesis discovery or education if that's a use case that's important. But most are doing prospective research based on this off-the-shelf data. So my team at Vanderbilt leads the Data and Research Center. So our job is to sort of harvest all of this information, organizing it, and then make it useful for researchers in a cloud-based ecosystem. We have about 20,000 researchers that have signed up, and with that, there's a lot of work that's going on. We have a really good problem, in that any given day there are multiple publications that are being accepted and published and indexed in PubMed, where people are using the platform of REDCap to do All of Us to do that work. So one of the things that we thought about was, can we actually sort of make the curation of those publications faster than having human level curation? And as part of the curation, that's is it an All of Us related publication, or is it just a mention of the program? If we find through this AI-generated curation process that it is a publication that used the platform or the resources, then we also do some AI work to catalog and to curate these into buckets or domains of interest from the scientific committee of the program. Things like rare diseases, common diseases, health equity, et cetera, or population health, et cetera. So we've developed this platform. It's purely automated at this point, and you can sort of see on the right-hand side, this is how it sort of comes out to the researchers and the community of participants if they want to know what's actually being done with their data in the All of Us platform. The second platform I want to mention is called Research Match. This one's been around about 15 years. We developed it at Vanderbilt, and then as part of a CTSA organizational platform. And it really exists just for a very simple purpose. We want to allow people that are out there in the community, anywhere in the US, to say that I'd like to participate in studies and trials. And then we'd like to make this matchmaking, this privacy-enabled matchmaking functionality available to researchers anywhere in the country where you've got participating institutions to match to those individuals, bring them in, educate, and hopefully include them in studies and trials. So this one's been going on for a while as well, and we've got over 17,000 researchers that have used it across 200 and something institutions. And we've got at least 1,000 publications. One thing that's really cool about that is if you're a scientist, anybody in this room would be able to look at those publications and say, this is fantastic, I see what's being done with that platform. But most of our consumers are not scientists. And so trying to figure out that translation from science language to lay language is really quite tricky. And we've tried it before and failed before because it's just the methods that we developed were just too human-centric and non-scalable. In this particular case, though, we did find good luck using generative AI, designed experiments, and got IRB approval to be able to sort of do this without a human in the loop, to basically generate these summaries of what's happening, what's the societal impact of what's happening with these publications, and again, make it available to those that are entrusting their data and their participation with us. So that's another area that's also publication focused. The last use case I want to mention is called REDCap. This is, REDCap stands for Research Electronic Data Capture. We created it about 20 years ago at Vanderbilt to support first the research enterprise at Vanderbilt, giving researchers an easy way to describe their study and basically have a data management system and platform underneath to help administer that data management collection and management plan. We've been very fortunate in this one. It's disseminated very well. We're in 8,000 institutions have licenses. We have many researchers in many countries. It's available at no cost to academic institutions, nonprofit and government organizations. One of the things that's really neat about REDCap, though, is that because it has that widespread dissemination, anything we drop into that platform has the ability to disseminate very widely, very quickly to the larger global research community. So as we were thinking about what we were going to do here first with generative AI, had some consultation calls with members of the consortium, and we kind of went through different ideas. And I realized that there are people out there that are not quite ready for this from an infrastructure standpoint. They don't have those walled gardens set up so that they can pass information along and be sure that their research-oriented data is not leaking into a computer system, et cetera. So there's some of that work that needs to happen at local institutions. And also you've just got differences of opinion on how comfortable people are with specific use cases. So we decided we'll put a minimum viable product together, and that minimum viable product will allow optional participation, both at the turn the whole thing on or off level within REDCap at a specific institution, or at the module level. And really we stuck to 3 really safe ones to get us started. We've got other ongoing work now with 2.0 in the works, but we released in January this 3 modules. One is Help Me Write. So that might be an invitation, it might be survey language, it might be preparatory information that you want prior to giving an e-consent form. And this one really is just like it sounds. While you're writing that, if you've got a rich text editor open, you can click it. It'll fix your grammar. It'll allow really simple prompts to make it less words, make it more accessible, change the tone, et cetera. The second module is around rendering case report forms and surveys in multiple language. That's a functionality that already existed in REDCap. This just makes it much easier to do those translations. And the third one is probably most interesting from a data perspective. Anytime we're collecting unstructured data, we have the ability now to have the researcher write a prompt or adopt a example prompt to be able to basically summarize that data. Again, we've got other things going, but that's what we started with. So the lessons learned so far are really to solve real problems. A lot of people can think about interesting ideas, and you can get about 75% of there by just playing around, but really coming up with real problems to solve and not just letting the technology drive has been really important. Think big, start small, and evolve, and especially in these first phases, do no harm. I love this quote. I'll just end with this quote in the bottom right-hand corner by Reid Hoffman, who started LinkedIn. If you're not embarrassed by the first version of your product, you've launched too late. So we really are trying to do that while also doing no harm. Thank you.
Thank you, Paul. Can I brag that I've been embarrassed many, many times? That's very good. All right, so as we had before, we're going to spend a few minutes with some questions. And actually, I wanted to honor Suchi Saria's preparation work and work from her notes in this part. But then we will have time for questions from the audience. And so just as before, the two microphones will be available to you when we get to that point. I had started off with these both good and not so good projections, and one of the not so good projections for the future was the possibility, maybe even likelihood of a bubble bursting in the biotech world. And I think, Fiona, you were actually hinting maybe a little bit about this, and maybe even internally within your own division at Novartis. So I'd like us all three of you to try to get real here. What really is different? What can we do now that we couldn't do before that you think is important? Or what do we feel like we're just on the verge of being able to do that we couldn't have thought of doing before? And just to be as grounded in reality as possible. I think Suchi is, if you know her, she is prone to asking very tough, pointed questions about this, and I think that was her intent here. So Fiona, should we check?
Okay, maybe I'll just start. I mean, I think some of the areas that I highlighted where I think we're already really seeing a difference. We are coming up with targets we never thought of. We are definitely designing molecules, even on targets that we've worked on for years. Generative AI is taking us into a completely different chemical space and finding chemotypes that have got away from past problems. And the safety is really, the predictive safety areas are really improving our attrition rate. So those are all good things. And I think they're really exactly that. And very AI-enabled molecules are now moving through. In fact, I would say most of our programs now use AI. So I think that's the good part. I think what's still difficult is the clinical translation. We are using AI to really accelerate how we do clinical trials, that in terms of optimizing the protocol, how we recruit patients, so that we think we can really speed up clinical trials, but we're still constrained by the length of the disease. What we're hoping for is more regulatory acceptance of biomarkers. That, I think, would be a real game changer. At the moment, if you've got very long sort of outcome trials, that's where everything can slow you down. So trying to find better ways of doing clinical trials and getting the readouts faster, I think, could be a major inflection point in how we can.
In the tech world, we oftentimes think in terms of tech stacks. And so maybe, the, let's say, binding prediction for designed molecules.
Yeah, that we're pretty good at now.
That's at the very bottom. What's next? What's next up?
Well, I think actually predicting the pharmacokinetics of what is this, how is this drug going to be metabolized, broken down, excreted. So definitely you can get good binding to the target, but what actually makes a medicine is a drug that can be taken orally, absorbed, lasts the right duration of action. That we're still really quite a long way away from.
Great. Lovisa?
So, yeah, first of all, it's interesting to reflect over that, 4 years ago, every sequence was not a structure. It was just not something that's something that is now so obvious to us that we can design binders relatively easy because we can actually fold a protein. So it has, these recent years have just made such huge advances. I think where I see where we are making a lot of progress, but I think there is an enormous sort of opportunity ahead, is really in agentic frameworks in our ability to interrogate what is already available as information around us. Coming back a little bit to your point with regards to how we can design in clinic based on competitor products or just known biology, and putting that into context, being able to drive it back to what does that PK/PD profile need to look like, really integrating quantitative medicine very early in your development. But also what we work a lot on, which I'm excited about, is how do you then translate it into how to actually engage with the target in order to drive that downstream PK/PD that you're after. Think checkpoint inhibitors, the real understanding of how you need to engage with your target in order to drive the clinical profile that you eventually are looking for, and how we can put such sort of frameworks in place a priori so that we are more validating our hypothesis as we go along. And I think there is a huge promise. I don't think we're there yet, but I think that it's exciting to see the advances that we're making in that more, I won't say autonomous science, but in some ways, very informed science, still with human in the loop.
I would say 5 to 10 years ago, technologists like me were very guilty of not respecting the incredible complexity of biology, especially human biology and the chemistry of the cell. And as we've come to terms with that, which has been slow in coming, I think we sometimes vacillate between thinking it's all too hard versus it's all solvable. And so I think, Paul, maybe you have the broadest view because you just have either direct or indirect contact with so many bioscientists around the world who are trying to get something out of data. What are you seeing?
I mean, I think for the last 20 years, I've said the same thing about my profession. This is the golden age of being an informatician. And I think it has been at the golden age. But I really think the next 5 years are going to be the golden age. And specifically, I think that because we now have this ability to democratize access to so many different solutionists. You mentioned earlier real-world data. We've spent a lot of time figuring out how to get REDCap to integrate well with healthcare systems. The next step of that would be, okay, you've got clinical notes. You don't necessarily need your own natural language processing expert anymore, or your de-identification expert, because maybe we can start using these tools for some of that work. So just across the board, I think democratizing access to really smart people yields pretty neat things that I know I've seen it many, many times that I would have never expected using these tools or platforms.
Yeah. So maybe I have a sense we've been on the optimistic note. So let's get into barriers here. So I'm skipping one of Suchi's questions here. What do you see as the technological, ethical, or cultural barriers that still stand in the way? And just to spark a few things that come to mind, will the FDA be able to cope with an exponential explosion of approval requests? Fiona, you mentioned about finding targets, but targets aren't patentable. So are there intellectual property barriers? Data, Paul, do scientists worry about credit assignment if they are willing to share data, or proprietary silos of data, Lovisa in the venture space? So if we go down the line, if there's one barrier that keeps you up at night the most in all this, what would that be?
I mean, for me, it's people who don't really believe or trust the technology, or they prefer to do it the way they've always done it. So I think getting people on board and really sort of believing and then trusting how and that's why I particularly gave that example of the cardiac safety prediction. If you've got a molecule
as a manager of premier biomedical research operation, how do you manage that?
I mean, I manage scientists, fortunately, so usually data is the best way. So I think actually demonstrating where AI is getting it right and how much time can be saved and what the benefits are, and really demonstrating, that's the key way of getting more people on board. And I think as we started to roll out AI, there was a reluctance for project teams to want to use all the tools, but the more they see those projects that are using them moving ahead fast, the more people are wanting to get access and use it themselves. So I think that applies to all of it. I think that the more people start to use it and not see it as a barrier and see it as a benefit to them, the better. And I mean, obviously people are afraid of losing their jobs, and that always comes up whenever I do anything. So that's always going to present a barrier for technology adoption. But when you can actually see, it could create more jobs, or it can make your job a lot easier so you could do more, then having that mindset can really help.
Lovisa, I think as my colleague Eric Horvitz would say, the number one barrier that we hear from our scientists is just give me more GPUs.
Yes, I think that's. I was almost going down that path, but I was actually more thinking about what you were saying with regards to culture and people, because I think that is this what Aviv alluded to earlier today on quality versus quantity, and that comes back a little bit to your sort of size question as well, because you also have the challenge still, I believe, between biologists and computational folks as to quality in very detailed data, as to quantity, and when quantity turns into quality because you can use AI. And I thought that was very well said, because I think explaining those differences has oftentimes, I believe, been one of the harder pieces when you're literally in the room of deciding on what you should do next and why. And it's easy if you're just sitting with a lot of computation people, because then it's all about more GPU and more power and more data. But I think it's still the balance in between, because we need really the deep understanding, because to your point in what might happen in 20 years, we can no longer even understand what AI is proposing to us. That's not where we want to be, and we can only sort of prevent that from coming by constantly keeping up on the biology side of things and understanding the complexities, realizing where a black box is, but then sort of narrowing down to what is the problem we really want to solve in that big black box. And that's a lot of culture in being able to have people from those different disciplines really come together and learn from each other in that aspect.
Maybe I have a question along this for Paul also, but just to be provocative, is it possible we're fooling ourselves? You know, Aviv, for example, in her lecture showed in some types of problems, if not approached smartly, would require just beyond astronomical amounts of data. Someone once told me that there have been fewer than 110 billion human beings, individuals of Homo sapiens, who have ever existed. So even if we had, say, genetic samples from all 110 billion human beings, would we have enough data to solve these problems?
No, and I think that's where synthetic data, I mean, really as to expanding your sample size and understanding, and I think leveraging evolution, and which is a very good way, I think, of being able to assess sort of many more variations that you would ever be able to do in the lab yourself. So I think that it's probably different from depending on which question you're asking, right? But it's an interesting thought, and I think we need to be open-minded of that. We will need to let go of the boundaries as to where we want to control and where we're okay with AI controlling, because you just want to get that sample space covered.
Paul, what motivates scientists? You could argue that a scientist is risking the future job prospects of their postdocs and their graduate students by joining all of us, couldn't you? You know, what's the motivation for participating in this?
As a participant or as a researcher?
As a researcher.
As a researcher, I mean, it's a phenomenal treasure trove of data that's already been collected. It has been properly vetted so that it's not considered human subjects data. It's not readily identifiable. So you could literally wake up at 2 a.m., have a great idea, and start working by 2:30 a.m. So, I mean, it's a phenomenal treasure trove. And I think what makes it that is the multimodal data elements.
So there's a quid pro quo. You divulge some of the data that you painstakingly have developed in your lab, but in exchange, you get access to everything. Is that
You don't have to, with the All of Us program, you don't have to, as a rank and file researcher, you don't have to give anything back.
Fantastic. All right. So now, in just a minute, we're going to open up to audience questions. And so there are the two microphones. But as we get ready for that, just go down the line. You saw my predictions. Is there one prediction that you wholeheartedly agree with, or you think is total bunk, that you'd like to call out, Fiona?
Can we flip back to it?
Yeah, if we could flip back to that slide, that might be good.
I can go first
Okay,
on
go ahead.
one of those. The 10-year you had with regards to no human in the loop or semi-automated, was it, yeah? I think I couldn't agree more. I think that's where we get to that super intelligence, where we can have closed loops of robotic and AI agentic frameworks working together and really propelling science forward. And that's something we're super excited about at Flagship as well with Lila and our initiatives in sort of polyintelligence, bringing together human intelligence and artificial intelligence.
Good, and you are allowed to tell me I'm full of stuff here.
We'll do that after. No.
Okay.
One that stuck in my mind is that AI discovery sort of outrun the ability of us to understand. But I think they've already done that, and but I don't find that threatening. I mean, it made me think about a car, and when I first was taught to drive by my father, he explained in great detail the combustion engine, and I could even look in the car and see all of the components. Now I look in the car, I mean, you can't see anything. It's just silver box. I have got no idea how it works. It doesn't seem to, the electric car doesn't work the same way, but I can still drive it just as well. So I don't think it matters that if we don't fully understand it, as long as we can use it to harness it to do the right thing.
I think you're psychologically much stronger than I am. Paul?
Yeah, just riffing off of that last comment. I mean, we've done a number of different real-world implementations, and the first version, that really far left skunkworks project, a lot of times they turn out awful, but the models are going to get better, and so a lot of times what we've done is just kind of kept working with that assumption. Sometimes we pit the machines against the machines to make more accurate predictions, but I think it's the case that it's going to continue to get better, and so design with that in mind.
All right, let's now open ourselves up to questions. We'll start here.
Thank you, Jeffrey Lieberman, Columbia University. I have a brief but multi-part question. So this is a technology for in silico drug discovery and design. Is it applicable to all types of therapeutic agents, biologicals, small molecule, RNA, et cetera? That's number one. Secondly, will it be applicable in terms of level of difficulty to the different disease areas or organ systems? And then the last question is, do you also expect that it'll be able to de-risk clinical trials once you get into the clinical development or the valley of death, with which a whole host of other factors can intervene?
Yeah, thank you for the question. So,
Maybe Lovisa start with the different modalities, because you're exploring.
No, I think that this is definitely applicable across the different modalities, as you alluded to. I mean, in basic it's chemistry. It's how we can understand whether it's an amino acid or a polypeptide in a protein versus base pairs. And I think most of the tools that we are applying can be made generally applicable in that sense. I think how to then apply them in different
Maybe
target
across
discoveries.
maybe across different disease areas. I mean, it doesn't equally apply across disease areas because certain disease areas are driven by single cell types. So obviously a tumor in the case of cancer or immunology may be B-cell driven or T-cell driven disease. It's easier to use AI to model a cell or a cell system or a pathway within a cell. I think where you have a very complex multicellular, multi-organ disease, and particularly if CNS, for example, where it's not at all simple to model as a single cell, that's further away from us to be able to use AI. And that's, again, why I use the mini organoids, because that is something we're trying to capture with these mini multicellular organoids that would give you that intermediate between the AI and then the more complex human system.
I think, in a way, Aviv's lecture sort of answers that question, that right now the limitations of AI are such that we have to take data from many different sources, including whole organs or even whole body data, and try to relate it to what we're able to infer at the molecular level. What we're not able to do right now, and we may never be able to, we don't know, is start from the molecular level and go all the way up. And until we get to that point, we're always going to have to be getting more specific to specific organs, specific conditions. Now over here.
Yes, I'm Carlos Arteaga. I'm director of the Simmons Cancer Center at UT Southwestern Medical Center in Dallas. So it's more of a comment than a question, but I did not hear a lot about prevention of and intervention against disease before we know it as it happens. And my question has a cancer undertone because of what I do for a living, but at least in cancer, we have increasingly, as we heard from Dr. Regev, single cell data. We have preneoplastic single cell data. We have gene expression from preneoplastic tissues. We have information on epithelial tissues that are at risk of subsequent transformation. Seems to me that with all that information and clinical information, population studies, we should be able to predict with precision and sort of hack Darwin and know exactly who is going to develop the disease, who is not. It's also likely will identify targets in those tissues that are not representative of the disease as we know it, where the intervention may be easier because we're targeting less complex biology than when the patients are diagnosed or when they have late disease. So I did not hear a lot about that, and I just wonder whether anybody can comment on discovery efforts in that setting.
It's a great call out. We really didn't address, let me call it just broadly prevention, but it's also related to all the advances in our understanding of the immune system, longevity, health span, and so on.
Maybe I'll start. I mean, there are certain disease areas where we are already doing prevention. I mean, cardiovascular risk reduction is the one that you'd call out. We can predict based on a whole load of parameters of lipid, cholesterol, family history, etc. But these are very, very long clinical trials, and a huge commitment. And so, oncology, I think, is one now where we have the potential to do preventative trials. Some preventative trials have been run, which have not been very successful. For example, the contribution of inflammation to oncology risk. But it's something. What we're trying to do is, there's all regulatory hurdles. You tend to start with the very late stage, third line, and then you go earlier. It's quite hard even to go into first line treatment, and try and work with the regulators to allow it. Of course, it requires a very, very safe molecule as well. But I think you're absolutely right. We are at the threshold now of being able to predict patients with different types of tumor types, and absolutely, that's exactly where we should go, because that's where you can get the most benefit.
Hey, Paul, you have a response, and we're going to go to the next.
Just thinking about all of us again, it's a great platform that has multimodal data, but it's a longitudinal data. So we certainly have people thinking about AI-generated foundational models with that data set. We have long-term data for people that register with EHR data. So we see a lot of people using it for replication studies, but also you could use it for your sort of frontline predictive medicine.
So next over here.
Hi, I'm Barry Kohler, physician-in-chief at Rockefeller University, and I first want to congratulate Paul. We've used Research Match and REDCap, and they are truly game changers that have profoundly affected translational research around the world. So, and looks like all of us is going to do the same thing, so congratulations. In terms of getting information back to people in language they can understand, one of the things that I learned from Vivian Lee was about making podcasts. And what I've been able to do is to convert some of my rather dense papers into, with one click, into a two-person podcast, people talking back and forth in lay language that anybody with a high school education would understand. And I think this is a game changer, and I've also done it for my informed consent form. But it also, I think, helps junior people hear language that they can use in their elevator pitch to actually get words that people who are not conversant in the jargon would understand. My real question, though, is really about number 2 on the right. I think everybody's been ignoring this, but that is the gorilla in the room, and I just saw in Science making AI to be able to predict who's going to be making bad things. And I served on an NAS committee for making countermeasures to biologic warfare agents. And I really think it's crucial that we have this discussion, but I think it needs to also be targeted to how we present ourselves on Capitol Hill with regard to why it's important to have science in this country. And if you don't believe in any other reasons, medical, humanitarian, or whatever, if you care about this country's national defense, you have to be committed to supporting biomedical science.
Well, thank you for saying that. And allow me to actually commend, there's work led by Eric Horvitz, who's here, on something called Paraphrase, and it really is trying to provide a scientific foundation for exactly that type of dialogue. And creating that, I think, in the world will be incredibly important. Yes, next here.
Hi, I'm Anwesha Day from Genentech. First, thank you for a really insightful session. I'm curious we've heard a lot about democratizing access, large amounts of data, how much is enough. One of the other things, especially in the drug discovery space, right, that we're grappling with, that in the face of sparse, publicly available data and everyone training on proprietary data, how are we going to think broadly as a community about benchmarking, right? All of our, whether it's agents, how is that benchmarking of the agents going to work? Keeping in mind the real IP, the intellectual property challenges for the AI drug discovery space. And given that we have everybody in the room and panelists from so many sectors, curious to hear your thoughts on how broadly, as a community, can we make better progress as we think of benchmarking?
Yeah, so I think we have opportunity for 3 great perspectives here, 4 if you include me, but from the big pharma, from the new venture space, and from the publicly funded space. What would create a breakthrough in access to data that could potentially really accelerate things? Fiona, do you have
I'm very open to actually sharing a lot of our what I call pre-competitive data. I think that benefits and lifts everything up. And I mean, this is where I'd really commend academic research for really putting so much data into the public domain very quickly. I think this is an extremely good way of operating in the AI area. And it's certainly something that I really try and encourage within Novartis or other companies that I've worked in. So the safety data that I talked about, I mean, we've actually done that through a partnership with a company, Deciphex, where we are giving them access to all of our toxicology data with a view of them developing the algorithms that will be broadly available for any other companies to use. Because for me, the value is not really in the data itself. It is, but the real value is in making better drugs that avoid any safety issues and that protects patients as well. So I think it's a mindset that as pharma we need to, and biotech companies, really we should continue to try and work together better to make sure we do benchmark the algorithms we're using. I'm sure the regulatory authorities, the FDA, if we come forward with models that we're using, are going to want to have those validated across the industry. So we can't, it's not in our interest to just try and take those forward on our own.
I think it's a quite complicated question, actually, because it's not just about making data available. It's making the right data available and data that is actually comparable. And oftentimes, data is generated under very different circumstances. And how can you actually tokenize data? How can you use federated learning to actually leverage data in a way where it can maintain some of its proprietary information that is needed for it to be useful? But I don't have a really good way of thinking about it, I would say, because I think it's easy when you have a small problem, but when you're trying to sort of solve it on a large scale, it's not so obvious to me how to do that in the best way.
I would just say from the academic medicine space, I think there's a big difference between sharing and sharing well, which is what you just said. Which requires a lot more time and energy up front, as you're planning your study and thinking about common data elements, et cetera. But beyond that, I also think, aligning the incentives, making sure that people are rewarded for their work sharing, and that there's some pretty good, governance and data use agreements in place, of course.
Let me say,
the best examples is the PDB, where everybody had to put up their protein structure under very stringent criteria and include all the raw data and analyze it in the correct way, and that's what enabled AlphaFold at the end of the day.
one good thing is, like the English language, which doesn't evolve or change very rapidly, biomolecules and proteins and human biology also doesn't change. So that does give us hope that if we ever do achieve significant foundation models of the sort, say, that Aviv had mentioned, then that would allow much smaller amounts of proprietary data to achieve very, very good specialized results. And so that may end up being the future as opposed to sharing. We're running short on time, but I see Victor's waiting, so we're going to take just 3 more questions, and I'm sorry you won't take those. So let's go here. Yeah.
I'll try to be quick then. My name is Jonathan Bean. I'm from Harvard Medical School in the VA Boston healthcare system, and I run a center on geriatric care. And my question relates to a lot of the discussions has been about a single disease model. Yet if we think about older adults, or I care for veterans, middle-aged veterans, you're dealing with multiple chronic conditions, multiple medications. And I'm wondering if you could just speak to the use of AI potentially for the later phase clinical trials, for thinking about how we simulate control groups and thinking about those sorts of challenges for these to get real-life solutions for promoting longevity.
Yeah, fantastic question. We were hitting on this about prevention before, but also things that happen with long-term chronic conditions. So on the clinical trials, challenges have been such a blocker. Will AI give us new ways to get over that hump?
Yeah, so I mean, one of the ways we are trying to address that, and this applies to all patient groups, whether it's elderly or women or even patients with genetic diseases, is really try to incorporate, first of all, real-world data into the databases, as well as patient insights and patient preferences in how you design the trial protocols. And the way that we're using AI is in what we call protocol AI. It's not a great invention of a name, but I mean, when we lay out how we recruit patients, we usually put in lots of restrictions or how we're going to do the trial. And then what the AI can do is then search against all the patient insights and the real world data to say even how fast, and previous trials, like if you include this parameter, will that accelerate the recruitment or will it actually slow it down? And also which sites around the world are likely to help recruit this type of people. So that's already being done and it's having really quite an impact, and you hope then that you can address the patient's resistance to why they wouldn't want to join a trial.
Hi, my name is Kira McHugh. I currently attend Howard University. So besides creating new drugs, what role do you think digital twins or cellular systemic metabolisms will play in advancing translational medicine and personalized patient care?
Lovisa, would you like to take that?
I think it's an excellent, that is going to be essential for everything we do in the future, I believe. In particular, the digital twins, I think, comes back a little bit what you were saying, thinking about how you can simulate biodynamic phases, and coming back to the previous question, how you can use that to simulate outcomes before you're actually standing in front of the problem, just will allow you to design your trials much better, to test your hypothesis much better. And yeah, I agree with you. I think it's an exceptional way of thinking about the future. Yeah.
Thank you.
All right, and now we have our last questioner.
Yeah.
Thank you. I'm going to be very brief. First of all, as you know, science is one of our strategies, 5 strategic goals, as you know. One, of course, is look at emerging science and technology. And here, there are really a couple of things I wanted to ask about. One, of course, is the cost of care. So the question posed to all of you is, will AI greatly reduce the cost of care eventually? But the more important question, Peter, is to look at your paradigm up there. You know, on September 17th, Stanford published a pre-pub paper in BioRx, in which they are able to use genomic design to form bacteriophages, functional, viable, and possibly would even kill bacteria. Now, don't you think your number 5 is going to accelerate a lot more? Because you can imagine that's a bioweapon that you'll be very concerned about. We've been concerned about synthetic biology and others, but this will greatly accelerate the problem.
Yeah,
Two
maybe,
questions.
On your first question, that's something that perplexes me because I think it's a given in the world of healthcare that prevention and early detection diagnosis should lead to lower burden and cost on the healthcare system. No, it's not. Right. Yeah, and the data would say otherwise. One of the things colleagues of ours at Microsoft had a sequential diagnosis paper, and the data shows clearly that as you give the AI more lab budget, it gets better and better diagnostic results. And so it begs the question, how much is enough? And if you put that power in every single human being's hands on the planet, do we just create a pandemic of worried people with dramatically increased burden on the healthcare system? And so I think the question is really open, and at the end of the day, it's not a technology question, I would argue. It's a policy question. The technology will be there. We'll be able to answer the question about prevention, for one thing. I think we'll have the tool. But if it, in fact, turns out to be the case that it doesn't in itself lead to lower burden on the healthcare system, but increased burden, we'll have to make some decisions as a society about what to do with that. Any final?
I think exactly as you say, it's like, what is the value creation and what is the value generation from our AI models and what it actually is to solve, where we oftentimes apply technologies towards the maybe not the most important problems. And I think that's a challenge for the equation as such. And we have the potential to point it in the right way, but we have to be very deliberate about how it's pointed to get the effect.
Let me give a chance for a last word from either Fiona or Paul to Victor's other question about the potential for real threats, bioterrorism or other biological threats.
I would just say thank you. I was sleeping too well at night. And,
well, with that note of gratitude, I think we'll end our panel. So thank you.
Thank you.